students. So for this lesson, we're going to start thinking about what happens to cells when they're ready to divide. Okay, so the process happens when cells get too big or when they need to essentially make a copy of themselves in order for reproduction. Okay, remember what some of the triggers are that help determine when a cell's ready to divide? Well, one of them is simply when it grows too big, then the surface area to volume ratio isn't efficient. And so the cell will make a copy of all of those organelles inside the cell, and then it will simply divide into two. So that's the trigger that causes that to happen. Well, the most important thing that needs to happen when a cell divides is that the DNA that's inside of the nucleus, those chromosomes, they need to make a copy of themselves before the cell divides, because every single cell that gets produced at the end needs to have the same copy of that DNA. So this is a process of which we look at the cell cycle, and the part where the DNA replicates and then lines up during the center of the cell and divides out is mitosis. So we're going to look at the overall process and then the specifics of mitosis. Okay? All right, so in your textbook, you can see this lovely little diagram here that looks at the cell cycle. It shows you that if you follow the timeline around, the majority of the time a cell is alive, it's actually just getting ready to divide. It's growing and it's doing the normal processes that all cells do, that normal metabolism, right? It's undergoing cellular respiration. It's providing energy for the cell. It's making proteins like crazy. And we've already gone over that process, haven't we? Okay. So most of the time, it's not concerned with doing anything it needs to get ready to divide until that little tiny section where cell division takes place. And then we're going to break that cell division part into the names of the stages. Okay? All right, so there we go. That's our cell cycle. The first part we're going to talk about is interphase, okay? And we'll do this on the handout as well. I'll just simply highlight some of the key terms here. So interphase, this is the resting stage. It's getting ready to divide when it needs to, but this is the normal process. The cell division part is actually called mitosis. Okay, so can you see here on this diagram here, you've got that getting ready to divide, and right before it gets ready to divide, the DNA makes a copy of itself. Every single chromosome makes an identical copy of itself, so then it's going to be ready to send the exact same information into two different cells in the end. Okay, so you start with one cell with information in it. How many chromosomes does a human have? We have 46. And so you've got 46 chromosomes. Each chromosome is going to replicate. And then in the process of mitosis, this is where every single chromosome will then split up. And one chromosome goes one way, one chromosome goes the other way. That happens for all 46 of them. And then you've got two cells at the end of this process where the genetic material inside of them is identical. Okay? Very complex process, lots of different stages and fancy names, but for grade 10, all we need to know is interphase, DNA replicates, mitosis, the DNA inside will start to move to the middle, it'll start to separate, and then the process by which that one cell actually cleaves off and produces two cells is called cytokinesis. All right, so there's a lot of terminology in this process. All right, you can see here we've got 3 chromosomes. Can you see that there's a red one, a blue one, and a green one? That counts as 3 chromosomes. Yes, you can see that there are, it's a replicated chromosome, isn't it? So the red chromosome, for example, looks like this. So we've already gone through the process by which one of these little red chromosomes in the diagram in the nucleus of that cell actually has already made a copy. Okay? Remember, this is still called a chromosome, all right? The 2 little sides of that chromosome have a particular name. They're called sister chromatids, and the sister chromatids are identical, okay? So you can think of this as maybe being chromosome number 1, and chromosome number 1 has now replicated in interphase. Okay, and then chromosome number 1 is going to have at some point the sister chromatids separate and create 2 different cells. I love the diagrams in the textbook here because you can see that the DNA, that nucleic acid, that genetic material has been dyed a certain color, but can you see that? You can see the outline of the nuclear membrane there. So this is the nucleus. This part here would be the cell membrane. And you can see that 1, 2, 3, there's 4 chromosomes right in there. The next stage, after they've sort of condensed and grouped up, well, then they line up along the middle. Okay, so how do we show that? Simply write the chromosomes right along the middle like this of the cell, and then there's some spindle fibers that are going to attach to the center part of the chromosome and pull them apart. Okay, we're going to go over the names of all of these on the next page. So you can see there that the sister chromatids will pull apart, and then they're starting to separate into 2 different cells. And this process here is mitosis. It finishes off at the end of mitosis with the last stage, which is called cytokinesis, and that's where the cell membranes pinch off and 2 cells are formed. And remember, the end of this process, each cell has the same number of chromosomes. Okay, so that's our textbook section. Let's have a look at part of the notes, and then we're going to go into each of the each stage in quite a bit of detail. All right, creating copies of a cell is important. Why? It's important for growth. It's important for repair, right? Cells need to make new cells every now and again. Happens all the time, actually. And that happens as your body gets older, as it ages. Cells are controlled by the DNA that's located in the cell's nucleus. And when a cell makes a copy of itself, the DNA must be replicated. And so an exact copy, okay, so let's fill that in or summarize it as you will. An exact copy is passed down to each cell that ends up with, and the name of those cells are called daughter cells. Okay? Daughter cells. This will ensure that that new cell has a complete DNA instruction set. Okay, so the cell cycle. As eukaryotic cells grow and divide, they go through 3 different phases. So I want you to think of this. There are 3 different stages. The first stage is interphase. So if we looked at that circle diagram we had in the textbook and compare it to here, here's where interphase would be. Interphase includes a G1 or a gap phase. This is where the cell is growing and doing all the things that it needs to do. There's an S phase, and then there's a G2 phase. And all of these together, okay, so I'm going to highlight those as a color. This grouping of those 3 is all interphase. The second major stage of cell growth is the mitosis stage. Okay, let's do that one in a different color so you can see. Mitosis, again, is this teeny little part of the entire timeline, if you will, of the cell cycle. And the mitosis stage has different phases. And this is where you have to use a little acronym to help you remember the different names of the phases. Okay? IPMAT, IPMAT, where you had your interphase, and then the mitosis is the PMAT part of that. So I have something called prophase, metaphase, anaphase, and the last one here is called telophase. And here's where you're going to come across a biology word where some of your teachers or biologists will call it telophase and some will say telophase. It doesn't really matter how you pronounce it. There's more than one pronunciation for that word. Okay? Once you finish that stage of mitosis where you have prophase, metaphase, anaphase, and telophase, so try to be familiar with all of those terms in the right order. Okay? The very last part of the cell cycle is cytokinesis, and all you do here is this is that very last stage of the cell cycle and the growth of the cell is where it divides into 2 daughter cells. Okay, so take a minute, get yourself caught up here, and then we're going to move on to having a look at the number of chromosomes. Okay, so let's look at that S phase of interphase. Here's what's going to happen. You need, at the very beginning, okay, I'm going to have two different chromosomes lined up right here. Okay, I want to show you what that looks like. Chromosome number 1, let's see. Chromosome number 1 looks like this. Why do you have two chromosome number 1s? For a second here, you want to look at this entire page and remember the fact that you have, as a human, you have 46 chromosomes. So you can diagram, you can write out this on a separate sheet of paper if you'd like, okay? 46 lines, you have 46 chromosomes. Remember, you have two chromosome number 1s. Okay, one came from the egg and one came from the sperm. The names we use for those are the maternal chromosome and the paternal chromosome. Okay, both of these are chromosome number 1s. Now, remember, that means that, for example, if your color of your eyes is written on chromosome number 1, you get information about your eye color from the egg, and you get information about eye color from the sperm. You get two chromosomes because you have two chromosome number 1s that are inside of your cell. Okay, so now this is considered a homologous pair. And again, you have two chromosome number 2s and two chromosome number 3s. Now, here's where it's interesting. During the interphase stage, remember, I've got two chromosomes here to start with. What happens in interphase? The DNA replicates. So remember, I have these chromosomes where I can easily show what that looks like. Here's a replicated chromosome number 1. Here's a replicated chromosome number 1. One's the maternal chromosome. The other one is the paternal chromosome. They are replicated chromosomes. Together, this makes a homologous pair. So let's just show what this is going to look like. So in the interphase stage, you have G1, you have S, and you have G2. When it's the S stage, that's when the DNA replicates. Now, how do we draw that? Simply draw it like this, where you've got a replicated chromosome, and they're held together by a center part. It's called the centromere. Okay, this again is a homologous pair. Okay, a homologous pair. And oftentimes it's kind of nice to color code them so you can see that this is one, this would be maybe the maternal one, and this would be the chromosome that came, or the paternal chromosome. All right, and during the phase when the chromosomes reach that phase in mitosis that's called metaphase, they actually do condense and become thicker. And that's when you can take a picture of them. So those cells that were in the textbook, and you can get some amazing pictures online, have a look at some of them, because it'll show you what those chromosomes look like. So visible under a microscope, obviously, but this is how we know what happens to the chromosomes throughout their stage. Okay. A few other details here. We're going to move on to, there's one more terminology here that we want to talk about. This is this concept right here of sister chromatids. Okay, and again, you've got a replicated chromosome. Because these two are identical, they are called sister chromatids. Okay, so again, this is called a chromosome. This is called a chromosome. This is a replicated chromosome. When does that take place? It takes place during interphase, at which stage? The S phase of interphase. Okay, this is homologous to this because they are both chromosome number 1s. Okay, this has sister chromatids, this has sister chromatids. You can see in the end, when I take this one cell that's going to have two chromosomes in it, at the end, I'm going to have two cells with the same number of chromosomes. And that is very important, okay? You want to have identical information in each cell. You want to have the same number of chromosomes to start with and end up with, because this is simply mitosis, and the whole purpose is cell division for growth and repair. Okay, I'm going to pause the video right here. We're going to do the first little bit of work, and then you're going to come back to the next video, and we're going to go through the stages of mitosis. So get ready to draw a little bit, get ready to have some color so you can add some detail to your diagram.